Hey everyone, I am here with Dr. William Walsh. He is a very famous doctor and PhD. He has treated thousands and thousands of patients in conditions like ADHD, behavioral disorders, autism, depression, bipolar disorder, schizophrenia, PTSD, and Alzheimer's disease. And he's developed very interesting protocols in determining how to treat these patients. He basically categorizes them based on a number of factors, and he's able to determine which group to put them in and give nutritional therapy based on that. So that's basically why I wanted to bring him on the podcast, so that we can talk about that. Tell our listeners a little bit about yourself, Dr. Walsh.
Okay, my organization is a not-for-profit public charity. We do experimental research. I'm basically become sort of a neuroscientist. My PhD is in chemical engineering, but I've never been a chemical engineer. But what I'm most interested in are we have one program where I have a team of experts that travel around the world training doctors, psychiatrists, and medical doctors in advanced nutrient therapy. We started with behavior disorders. I have probably the world's biggest chemistry database in behavior, and also very large databases for autism, depression, schizophrenia. And from this, I've also worked directly with 30,000 patients. And through this, we've identified probably the 6 or 7 leading nutrient imbalances you find in mental disorders. And we've learned that if we simply do lab work and medical history to identify the imbalances and normalize those 7 critical factors, that we've had really great success with violent behavior, ADHD, even autism, schizophrenia, and we published a lot of this at national meetings. We have some nice journal articles, but basically we're on a mission really, because we're finding that we can help people who have serious mental disorders without having to resort to drug medications. Drug medications have helped a lot of people, but I think that there's all this momentum to continuing use of these really powerful drugs. I started many years ago as a scientist working in chemistry and nuclear fields. And I was a volunteer as a young man, volunteer at a local penitentiary, Statesville Penitentiary. And before I knew it, I had a group of 125 volunteers. And what I learned was after I started meeting the parents that had a child that became a criminal, I learned that many of them were like that from the beginning. I began to suspect they had chemical imbalances. We started doing lab work testing. I was at a national laboratory with great chemists. And anyway, I found out that, became convinced that people who have severe behavior disorders are born with that predisposition. And so that started it. We've now worked with more than 30,000 patients. I have huge chemistry databases. And we now know we've developed a system for relatively inexpensive lab work, blood and urine testing, a medical history system where we can identify and diagnose the major chemical imbalances in these disorders. Methylation, overmethylation, or undermethylation. Copper overload has a massive effect on norepinephrine and dopamine. We've learned we really focus on the neurotransmitters and how we can adjust them and normalize them with nutrient therapy.
Okay. So first of all, are different conditions treated in different ways, or do you have a general template for every condition?
Well, it's really, the testing may be different for different conditions, especially autism and schizophrenia are somewhat different. But basically, we're trying to identify which neurotransmitters are misbehaving. And then we're trying to find out why by doing the lab tests that we do. And sometimes we can identify why. For example, if a person's undermethylated, that's associated with low serotonin depression. If people have classic paranoid schizophrenia, that's associated with hyper dopamine activity. So, but we've now learned, especially with epigenetics, we now have epigenetic therapies that we know how to regulate enzymes and we know how to change gene expression. And we're able, therefore, to correct, you might say, genetic or acquired abnormalities in neurotransmission.
Okay, so let's take, for example, depression. If someone has depression and they go to you, what's the first thing you're going to try to figure out for them?
First thing we're going to do is try to figure out what phenotype of depression they have. I presented this at an annual meeting of the APA not long ago. We've discovered with this gigantic database that we have that depression is a name given to 5 completely different conditions with different neurotransmission abnormalities and needing different treatments. And so what we would first want to do is identify, do they have the undermethylated version of depression? Do they have a metal metabolism disorder involving mainly copper and zinc? Do they have an imbalance related to a pyrole disorder? So that's the first thing we would do. Once we get that identified, then we study the individual nutrient imbalances associated with those problems. And we have therapies that can, at least they attempt to, and I think they can, in most cases, normalize neurotransmission. And we've done a number of outcome studies. We've published many studies, including a peer-reviewed study just in the last few months. And the outcomes are really very good. They're quite exciting. I think they're better than anything I see for psychiatric drugs.
Okay, and when you talk about epigenetics, are you mainly referring to methylation, or you're referring to maybe acetylation as well, or other factors of epigenetics?
Well, it has to be both because it's really a competition between methyl and acetyl at these CpG islands on the DNA. And so we've learned that it's not really the amount of SAMe methylation capability or the acetylate or the coenzyme Q10, the acetyl coenzyme Q10, it's the enzymes that control. So we're focusing on the deacetylases and the acetylases and the methylases, and we've learned how to do that rather well. I've been focusing on this for more than 10 years. If you increase methylation, you tend to shut off or lower the rate of genetic expression. And so what we've learned 30 years ago, that it's not the amount of serotonin a person's got that matters with their mental health. It's the reuptake, the rate at which the neurotransmitter, in this case serotonin, that was ejected into the synapse, how fast it goes back to that original cell. We've learned that in 1985. A lot of nutritionists don't seem to realize that. So the focus is on there is something called a transporter called SERT, S-E-R-T, genetically expressed. It goes on, it's on the membranes of these presynaptic neurons, and they're really it's a passageway for the passageways for the serotonin to go back for reuptake. So the dominant factor is reuptake, and that overwhelms the effect of methylation. And you need to look at both of them, and that's what we do. I think we're very good at that. And you have to do that or you get the wrong answer. I just had a patient today who found out they were undermethylated. They found they had MTHFR SNPs and believe that they are undermethylated. I think they are undermethylated, and they were getting worse on folates. And so I explained to them why. Really, the important thing is reuptake, and we now know with nutrient therapy how to adjust reuptake, which dominates serotonin neurotransmission.
So how does that affect the reuptake if someone is increasing methylation, let's say?
Okay, if you increase methylation, what are you doing? You are shutting down the genetic expression of these reuptake proteins. So you're getting fewer passageways for reuptake. And so what happens is that you're trapping, just like it's a natural serotonin reuptake inhibitor. It does the same thing that SSRI antidepressants do, Prozac and Paxil. They do it by a different way. The way those drugs work is they go directly at these reuptake proteins and they disable them or they weaken them. So the serotonin gets trapped longer in the synapse. What we do is we can lower the population of them by increasing methylation, and it works really well. It takes usually about 4 to 6 weeks. Our treatment doesn't work as fast as a drug, but it does it naturally without side effects, and it does it just as well.
Interesting. So the question is, so you're saying that people with, I'm assuming you're not a big fan of the serotonin reuptake inhibitors, and
I am. I've met, you know, I've met hundreds of people who said that these things save their lives. But I think it's an expedient. And now that we have more knowledge of how the brain works, better neuroscience, I think this is going to fade away as science advances. And I think the drug companies are trying to keep it the way it is. I mean, there's a huge amount of money. These are all billion-dollar drugs, and I don't blame them for trying to keep their bottom line good. But there's very little research aimed at doing this naturally. And even though now the neuroscience is advanced, so we know how to do a lot of it naturally.
Yeah, I mean, so I still want to stick on this topic because it's an interesting one where I get a lot of people who will be like, you know, I do well on methylation, and then a lot of people who say this is terrible for me, right?
Exactly.
And I think a lot of it has to do with the brain. It's definitely messing with some neurochemicals in the brain. So what I want to figure out is, so you're telling me when someone is methylating, the more they methylate, the less proteins like SERT, which is that serotonin transporter, will be produced. And
in general, it does that, yes. In fact, it tends to lower genetic expression across the board. But we have to focus on what's the major dominant factor in depression. In this case, a low serotonin activity depression.
So, for example, if you increase SERT, that will increase serotonin within the cells?
If you increase SERT, it's going to escape out of the synapse back, and you're going to lower activity in the synapse. You want to trap the serotonin in the synapse. So
then if you do methylate, then that's going to lower SERT, and that will increase serotonin in the synapse.
Yes, it will. Exactly. They won't have as many of these passageway escape routes from the synapse back to the original neuron.
So you're saying if someone is low on serotonin, it would be good to methylate.
Yes. If they have a mental condition, whether it's depression or anxiety or whatever, if the dominant reason is low activity at serotonin receptors, yes. And the nice thing is we now have a system where we can be 95% accurate, roughly, of identifying who needs that and the patients who might need folates. There are patients who do beautifully on folates because folates are natural serotonin and dopamine reuptake promoters. They elevate, you understand? They elevate the genetic expression, and so they knock dopamine down. If you give somebody folates, it's one of our major therapies for people with severe panic disorders and anxiety disorders. If they have really high norepinephrine and dopamine activity, it works beautifully.
By decreasing it. So let's say, is folate going to increase or decrease serotonin in the synapse?
Folates work really hand in hand with acetylation. They promote acetylation. To get technical, they disable the deacetylases. So they tend to help the acetyl vis-a-vis the methylation and that competition between methyl and acetyl that at DNA, which really sort of dominates the expression. Yeah.
Okay. So, but what I'm trying to figure out is, when is it good to take folate?
It's good to take folate.
If someone is high on dopamine?
If you have not the amount of dopamine, but neurotransmission of dopamine. Again, that is not related quite so much to the amount of dopamine or serotonin. It's really the reuptake. And dopamine is a little bit more complicated than serotonin, but if a person has too much dopamine activity, they're prone to anxiety, they're prone to panic disorders, they're prone to paranoid schizophrenia, they're prone to attention deficit disorders. And so what we would do is first, with our lab testing and our medical evaluation, I think we're very accurate and good at identifying which neurotransmitters are misbehaving, and then we act accordingly.
Interesting. Okay, so how would you know, for example, based on your test, which test would you use if someone is a high or low methylator? Would you use homocysteine? Would you use the serum levels of B6 or B9 and B12? What tests do you think are the most important in determining if someone is a high or low methylator?
Methylation is really important, and because that involves also epigenetics, and there are lab tests that I like to use. For years, we've used whole blood histamine. The reason is that histamine is regulated by methylation. That's how the body regulates the amount of it. So if you're undermethylated, you'll tend to have sky-high whole blood histamine. However, there are flaws and difficulties with using this. It's affected by antihistamines if a person's taking them, and then the other tests we like to use. We don't test for folate directly. We did that. I've tried every test you can imagine over the years, and so we've learned a nice protocol of lab testing. We have to test for serum copper and serum ceruloplasmin. At the same time, plasma zinc, not serum zinc. And that's because that has such an important effect on neurotransmission. You need zinc to help make GABA, for example. And if your copper is abnormal, which is not too often, we have a system in our bodies that regulates copper really nicely, even if you're chewing on copper bars all day long. If that system's working, your copper level will still be fairly normal. But if your copper level, if that doesn't work, if you've got SNPs and you've got mutations, and you don't have the system that regulates copper and it goes up, well, that's a recipe. That's a recipe for disaster. That knocks your dopamine activity sharply down, dramatically down, and your norepinephrine up. So we have to do metal metabolism analysis, which is fairly inexpensive and easy to correct. I was talking to a doctor just yesterday who has a patient with shockingly high copper levels, and the symptoms are exactly what we would have expected. We now know that that's the major cause of postpartum depression, is a woman after a pregnancy, during which time the copper level for the fetus has to be more than double for the mother. They don't have the capability of bringing it back down to normal after delivery. So that's another example. We do a Pyrole test. Are you familiar with Pyroles?
Yeah. Okay. So yeah, let's talk about these blood markers. I actually have a company, Lab Test Analyzer. We test about 200, we don't test them, we analyze about 250 biomarkers. And some of them are the ones you mentioned, like serum copper, zinc, ceruloplasmin. I don't think we have cryptopyroles just yet, but we're basically just like giving information when someone uploads it, telling them what the optimal ranges are.
That's great, especially the ranges.
Yeah, because basically when you go to a doctor and you get various tests and it's like, okay, this is normal, but actually if you look in the research, you see that what they consider normal and what's optimal is very different. There's a lot of research on optimal ranges for various markers, and so actually, that's why I want to talk to you about these 4 markers here. You got cryptopyroles, serum copper, plasma zinc, and ceruloplasmin. So let's start with cryptopyroles. What exactly? Can you explain what is a cryptopyrole?
Sure. Cryptopyrole is involved in the biochemistry and the bone marrow and the spleen. It has a lot to do with the formation of hemoglobin and some of these important blood chemicals, and really it's a byproduct. In other words, it's like A plus B gives hemoglobin plus a pyrole, and the pyroles have no value in the human body. They're quickly cleared out of the body through the blood and through the urine. And some people have, because of genetics, or probably because of genetics, this runs in families, they have far too much of it, maybe 5 or 10 times too much. And that's a recipe for dramatic zinc and B6 deficiency. And they have all the symptoms of zinc and B6 deficiency.
The issue I have with a Pyrole test, and again, I think you're doing it based on your clinical experience, but there really isn't a lot of science that you can look up on cryptopyroles. Like you can just go to PubMed and type it in. There's really not a lot of research that comes up.
There's not a lot, but there's a lot that is now going on underway. And there are a couple of review articles done by Dr. McGinnis. I was a co-author of this, and we have more than 80 or 90 journal references, including a lot of peer-reviewed journals. So there is some there. Okay. It's kind of, it is frustrating for a lot of our doctors because they like to collaborate with other doctors and explain to their patients. And there's really not enough information, but it's growing rapidly. But it is a disorder that is real. It does exist, and it can have a major impact on people, especially if a person has a behavior disorder. Just today, I heard about a 5th grader who was extraordinarily violent and is attacking his other students and destroys things. And if that child happens to have pyrole disorder, he will probably be better within a month. And because that is a disorder that has to do with the with biochemistry in the spleen and bone marrow, and some people create too much of this byproduct of these reactions called pyroles, and they strip zinc and B6 out of your system. And B6 is necessary for synthesis of serotonin, dopamine, norepinephrine. B6 is incredibly important. And usually a person gets all they need from their diet. But if you got pyrole disorder, it's going to strip it right out of your bloodstream. And,
So
and I think you're going to want to use that test once you get experience with it. It only happens, I think, in the general population, maybe 10% of people. But in mental illness people, especially behavior disorders. And now we know it's more than 38% of autistic children have pyrole disorder. It's also one of the major causes of depression, but it's only 17% of depressives have that form of depression. But if you have severe pyrole disorder, the reason might be just because of the pyroles.
So if you have high cryptopyroles, would you notice any difference in your zinc levels? Because you're saying the reason why cryptopyroles are bad, this is a metabolic byproduct of various factors, and basically the reason it's bad is because it'll lower your B6 and your zinc. So
one thing that would be really helpful, or the first thing, is that the symptoms and traits of a person with pyrole disorder are strikingly obvious. And we can actually diagnose a person with pyroles and predict their lab result before we've done the test, because the symptoms are just so striking. These people are not hungry in the morning. They're night people. They have mood swings that are striking. They're famous for their temper. They can't tolerate. They sunburn rather than tan. On and on.
But that could be from like basically just neurotransmitter imbalances or 20 other causes. I mean, there's a lot of genetics with various receptors and dopamine and serotonin, like you mentioned. The question is, how do we know that if someone has higher cryptopyroles, that it's bad for them? So the question is, can we measure B6 and zinc in order to do that? Or since we can't measure it enough, you're saying if they have high cryptopyroles and they have these symptoms, then we should just treat them with B6 and zinc rather than checking those lab markers.
No, we feel that the medical history and the symptoms and traits are a key part of this. So what we would want for that case, if we get high cryptopyroles and have the confirmation by unusually low zinc levels, and if they have the classic symptoms of a pyrole disorder, which are striking, then we're confident. But even then, we would be very careful to review them and make sure that this is you never know if you've got some people have 3 or 4 different chemical imbalances. You never know which one's the dominant one, but pyroles often are the dominant problem for some people. And
okay.
So medically,
but you would see that in their plasma zinc, though. You mentioned that serum zinc is not good, but plasma zinc is. Yeah. So if someone has high plasma zinc and let's say or higher or normal plasma zinc and higher cryptopyroles, would you still treat them with zinc?
Well, I would know for sure if that happened. One of the 2 labs was not reliable, and this plasma zinc is a problem because many labs and even some of the best labs like Quest or LabCorp, they're supposed to take it in as plasma, but actually serum zinc is almost identically the same as plasma zinc. The problem is cross-contamination. This has been a nagging problem for us a long time. If we get a person with high pyroles and high zinc, the first thing we suspect is that the zinc lab wasn't done right, and we ask them to go back and repeat it. And usually that gives us the answer. Having the 2 together is incongruent. It almost never happens.
Interesting. And so why, I mean, the serum zinc is a much more common test rather than plasma zinc, in my understanding.
Absolutely should not be. Serum zinc is a terrible analysis because and I've done 30,000 or maybe probably 50,000 zinc assays, and what plagues us is if somebody accidentally does a serum, you're going to get crazy levels, and they're going to be generally a lot higher than reality. Cross-contamination is the reason.
Interesting. Why is the plasma more accurate than the serum?
Because it doesn't have the cross-contamination problem. The tubes for plasma are vacuum outgassed. They're vacuum outgassed and acid etched to remove any traces of zinc on those tubes. And then the stoppers
so you're saying the tubes might be contaminated with zinc or like small traces?
That's the reason. That's the only reason. The cross-contamination from the tubes, that's the reason. And
so when they're checking plasma, they decontaminate the tubes better?
Absolutely. And please never use serum zinc and rely on it, because you're going to get a lot of crazy high levels that are not real.
It's never lower though. If someone has low serum zinc, what would you make of that?
Well, they're certainly low. It might be lower, but you don't always get cross-contamination from the zinc tubes, from the serum tubes, but it happens so often that it's been plaguing us for 30 years and still is plaguing us.
But you would never have a false low level, meaning you're saying like these tubes can be contaminated with zinc, like small levels of zinc. So if you get a serum zinc, then you might have very high levels of zinc or whatever, or inaccurately high levels.
You're absolutely right. If it's low, you're right. If it's low serum zinc, it's low.
Right.
You're absolutely right.
But the question is, I've actually gotten a zinc test. And I came out with low serum zinc, even on their ranges, believe it or not, right? Now this was odd because I eat meat I'm basically like on a meat-based diet. And so I eat meat most of the day. I take about 15 milligrams of zinc a day. And my diet must contain about 20, 25 milligrams of zinc a day. So the fact that I had low serum zinc was very odd.
Genetic.
You think it's just genetics?
Well, I know it is. It runs in families. When I was bringing criminals to Carl Pfeiffer back years ago, if you know who he was collaborating with me in the early days with these violent criminals fresh out of prison. He made me go through the lab work. He said I don't like blood draws, but he made me go through it. Don't say no to him. And he found out that I am extraordinarily zinc deficient. I've got two brothers that are more than 6'3", and I'm 5'8". And what he found out I was terribly zinc deficient. We repeated it a few times. My diet is great. I think I get at least 15 or 20 milligrams from my diet. I take 100 milligrams of zinc a day. And,
oh my gosh. Wow.
I'm so zinc deficient.
You take that with copper?
My copper level was okay.
So you don't take it with copper. I mean, there's some people who take zinc and then they become deficient in copper. If you take more than, let's say, 30 milligrams chronically, and this is not like a heavily genetic disorder, then you're also going to be deficient in copper. So that's why I'm curious if you don't take copper with that.
Not unless I needed to. If my copper level was low and I had low copper and low zinc, I'd take both. However, what does zinc do? What controls and regulates copper and zinc is a genetically expressed protein called metallothionein. Metallothionein. And it turns out that the way to if you've got a SNP in your metallothionein, and there are major SNPs that can really cut that down, and that's a major reason why some people are zinc, I think that's why I'm zinc deficient. I have SNPs in that enzyme, and so I'm fighting genetics. So what it amounts to is that zinc actually is the primary epigenetic agent that increases the function of metallothionein proteins. So if you're low in copper and low in zinc, many people, if it's because of metallothionein, like in my case, you tend to have both of them normalize. Copper gets better, even though
because the metallothionein helps with the absorption of copper as well.
It helps with the genetic expression of the protein that regulates it.
Yeah, I'm saying, and then if you have higher metallothionein, that helps regulate your copper better as well?
Oh, absolutely.
Okay.
Yeah. I was born with poor metallothionein function, and as a result, I'm genetically predisposed to very low zinc. Fortunately, I found that out. I think I'd be dead by now because of all the terrible things that happen if you're really terribly, severely zinc deficient. I've been doing this for 35 years.
So you think the zinc deficiency as a child stunted your growth?
Yes. I'm sure.
And what is that because of? Does it have anything to do with growth hormone or like?
Absolutely. That's been known for 50 years that zinc deficient people tend to have their growth stunted. I had a college student who was 4'9", a brilliant young girl, But we found she was strikingly zinc deficient. We gave her zinc. She was in college. She was in college at Notre Dame. She's now 5'2".
Oh my gosh.
Yes.
Wow.
And this is really great if you've got, say, teenagers, or if you've got children who have behavior problems or ADHD. We can tell them the truth that if they happen to have a zinc deficiency, they will probably become the height they're supposed to be. It'll probably make them bigger. And actually it affects muscle development. So it helps us motivate, others who might not want to comply. But it's true. It's real.
Wow, that's incredible. So the question I have about copper, though, and this is, the question is copper in the blood stable? Serum copper, ceruloplasmin. Ceruloplasmin is also an inflammatory marker. If you have high ceruloplasmin, it's an acute phase reactant, correct?
It's an acute phase reactant, and it's what's known as a dismutase. And when they say acute phase reaction, they used to think it was the copper that was causing the problem. The reality is, if you have inflammation, the body will genetically express more ceruloplasmin, because that's one of the ways you get. It's one of the ways you get rid of the oxidative overload. It's a dismutase. It can do things that glutathione cannot do. And so it's the fact that it's higher where there's inflammation doesn't necessarily mean that it causes inflammation. And I think it doesn't.
So you just think it's correlated with inflammation, but it's actually it's a good protein that can be helpful in various ways.
It's the body trying to cope with inflammation. Yeah,
right.
We have an amazing system where when something goes wrong in your body, genetic expression will adapt and change and try to fix it.
Cool. What kind of genetic program do you use? By the way, I'm not sure how familiar you are with Self-Hack, but I mentioned Lab Test Analyzer, and then we also have a genetic program called Self-Decode.
Yes, I'm familiar with them. Okay. I think that this is the future, frankly, but it's not quite ready for prime time for a lot of doctors who don't really get it all. There's a lot of, quite a few of my friends, in fact, maybe even half of really well-known nutritionists who try to evaluate methylation based on genetic tests together with the methylation, the whole methylation system. And this would be fabulous if they not only tested for the SNPs like MTHFR and others that would tend to lower methylation, but there are SNPs that tend to increase methylation. And as I said, there are 8% of mentally ill people have overmethylation. They have a glut of methylation because of genetic SNPs. If your genetic system would actually evaluate them and evaluate them together. See, it's like a tug of war. You've got SNPs that tend toward undermethylation, SNPs that tend toward overmethylation. Question is, who's winning the war? You cannot tell if a person has a 699T homozygous. I'm sure you know what that means. That means they're probably 70% likely, at least, that they are undermethylated, but it's not 100%. I've had people with that homozygous SNPs were overmethylated because there were SNPs that were causing the overmethylation that were more powerful. But it has to do mainly with the SNPs involved in there's about 80 or 90 really important methylation reactions. Only 2 of them consume about 90% of all the methyl. So if those reactions are inhibited, you'll wind up with a glut of SAMe, and you'll be overmethylated, even though your methylation system's working well. It might work too well.
So I actually 100% agree with what you're saying. You're basically saying that, okay, so the human body has 10 million SNPs, right?
That's how many they've found so far, right?
Right, that's how many. If you have MTHFR 699,
yeah. Or 1298,
if you have that main MTHFR SNP, you don't know about the other 100 SNPs that also have to do with methylation.
Absolutely. One great way of doing it is to look at homocysteine. If homocysteine is high and you got the SNPs, then you really need to address it and do something there. However, if you've got the SNPs, even if they're homozygous, and your homocysteine is normal, well, why do you need that methyl part of the methylation cycle? It's really to convert the homocysteine back to methionine. So it's more than just looking at the MTHFR. There are so many people out there, so many doctors even, who if they see the MTHFR SNP that's serious, they will immediately throw folates at them, especially methylfolate, wow, that'll be great for a lot of them, and it's going to cause terrible worsening in others. And we know which ones they are. Our testing and evaluation can identify who needs it and who needs to avoid it like poison.
Yeah, I mean, that's really interesting. So when I was looking at these various programs, before I started Self Decode, I was basically like, all of them had about like 20 SNPs, 30 SNPs, this and that. And that's something that you're mentioning. Like, you know, if you just look at the MTHFR, that's a problem. So what I did was, we have a database of 50,000 SNPs, but then
That's
now
better.
the problem, yeah,
That's better. That's a lot better.
now the problem is that a lot of people are confused. Like, hey, I looked at this SNP and this SNP says one thing, and then you have these 5 other SNPs that says something else. What's the deal? I don't know what to make of it. And
the deal is compensation. The human body is quite amazing. And there's all the, you know, first of all, methylation. That whole methylation profile is only less than 5% of human biochemistry. And if something goes wrong in any part of your methylation, there are other parts of that system that will compensate. They will compensate. And so you might have a SNP that looks really awful, and it might have absolutely no effect on a human being. Or it might have a terrible effect on a human being. Could go either way.
Yeah, that's something that people don't realize. I mean, they have, there's just so many SNPs involved. And what we're trying to do is actually, we're trying to work with whole genome sequencing partners, and also update our database to have 150,000 SNPs. And we're also trying to compute the data to give people an overview of how they are in like the big picture. I mean, that I would say is the very hard part, is to combine all the information in the SNPs or other parts of the genome, and then give people a big picture view of what's going on.
There's a group of people in England, young people, who are doing exactly what you're saying, and what they're doing is getting all the SNPs, the entire genome, and they're using artificial intelligence together with diagnosis of disorders, and they're just beginning. But to look at the SNPs really involve just the structure of the genetically expressed proteins. The SNPs tell you what is being produced genetically, and if you've got SNPs, what is not being produced quite right. It has nothing to do with epigenetics. Epigenetics has to do only with the rate at which these proteins are produced, whether they're okay or whether they have SNPs. And the two factors must be put in together, and eventually somebody's going to put together a really neat program for epigenetics as well as the genetic studies. And I think that's the future. I think that's going to be a marvelous way to accurately diagnose people with all kinds of disorders.
The issue with the epigenetics right now is it's just too expensive to get those tests.
Not necessarily. For example, all we have to do, if we find a person, if we can diagnose undermethylation, we can help people who have low serotonin illnesses. And so it's not expensive at all for us. All we have to do is identify which neurotransmitters are abnormal, and we know which nutrients tend to boost or lower neurotransmission. Different for each neurotransmitter. We can do it now inexpensively.
So what other tests do you look out for, like lab tests, besides the 4 that we discussed?
Well, we certainly want to get a complete picture of the person. And so we want to know their liver function, for example. We want to know their electrolytes. And so we probably have a total of maybe 90 different lab tests that we do. Sometimes we'll do different tests for different people. If we have a violent child, we might want to do a hair analysis, because that has special ability to identify children who are prone to really severe violence. We like to do CRP. I think one test that's going to be really popular soon is 8-oxylguanine. Are you familiar with that one? Is
that the one that measures DNA oxidative stress?
Absolutely. Guanine is one of the 4 nucleotides in DNA, and we know it's the weak link. It's the one with the highest charge. It's the one that's most vulnerable to oxidative stress. And we've got a lot of DNA in our body. Really, we have 30 trillion DNAs. We have 37 trillion cells. Not all of them have DNA, but 30 trillion of our cells have a DNA. And so if you get excessive oxidative overload, what happens is the guanine gets oxidized, and there's a couple of, I think there's 3 tests right now that are not expensive, where you can actually determine whether or not you're close to a breakdown. I think that's I think we have the ability now, and will have the ability to predict and prevent autism and these epigenetic disorders. The major epigenetic disorders, we know that cancer is epigenetic. And I don't understand why no one is looking at prevention of cancer, because all you really would need to do in a checkup is check out the amount of guanine oxidation, and you can see whether you're on the verge of an epigenetic onset of cancer. We now know that cancer is an event. It happens suddenly. It's the creation of a cancer stem cell. And we know that clearly, and yet everybody's trying to, all the research I see is aimed at allowing cancer to happen and then using these billion-dollar medications and treatments to try to, cope with such a complex disorder. And the epigenetic disorders, I'm convinced that it includes schizophrenia and bipolar, certainly post-traumatic stress, autism, and cancer. And some people is starting to look like many forms of heart disease are epigenetic. So that's, I think, a movement in health that is just going to be on and on. And but your testing profile, if it included some of those tests, you not only could tell them how to deal with disorders, you could show them how to do a good job of preventing them.
Well, I actually think we have the oxyguanine one. We do have about 250 markers, and we're adding them every week pretty much. And it includes the whole spectrum of the liver test, the whole metabolic panel, everything. And then it has a lot of these less common ones. I think, yeah, we do. We might even have cryptopyrroles. I'm actually not even sure because we add them every day. So I don't know if we got to that one yet or not.
It's a difficult test. It's a urine test, and the problem is that the pyrrole molecules tend to disappear and dissociate quickly. So the challenge is to get the urine sample to the lab in good condition. So that's the biggest problem with it.
So when you see a patient, are you giving them like 96 labs? I mean, they're paying for most of these labs, correct?
Yeah, and we've now got a standardized profile and a lab that will do these labs very accurately, and for about $350 to $400, all of the labs.
$350 to $400 for all 96 labs you mentioned?
Yeah, and a lot of it, I mean, I don't know how familiar you are with cost of labs. You probably know a lot about it, but it was an education for me when I started doing this. For example, whole blood histamine, okay? It used to cost, 20 years ago, it would cost $115 for that test. Well, once our clinic got really large and we started seeing thousands of patients regularly, they came to me and said, hey, great, we're able to reduce the cost down to $95. Well, we now can get that test for $12.
Wow. Is that because you have volume?
And they're making a profit on it. Zinc, if you wanted to do a plasma or serum zinc and just ordered that separately with one of the major labs, it's probably between $60 and $80 for that test. We can get zinc, copper, lead, total $12 for all three. And they're making a profit on it because it has to do with the volume. It's not the cost of the test, it's the volume.
So when you test, do you test serum levels of heavy metals? Those are not very accurate.
They're not reliable, and heavy metals tend to disappear from blood really quickly. The exception is lead. Lead tends to linger longer than the others.
So you test, you test serum lead.
Whole blood lead is good. Actually, analysis of lead is even better.
And so how are you testing the other heavy metals? Are you using urine or hair analysis?
We don't directly test for them usually. I've done a lot of experimental work with them. I was really concerned about the amount of mercury, for example, with autism and whether they tended to have more mercury in their bodies and in their brains. And there are special tests we've done for that are not really quite commercial. There are a lot of challenge tests people are trying to do where they'll test, maybe do a 24-hour test of urine, measure the amount of lead or mercury or cadmium or whatever you have in it, and then they put in they'll give them a strong antioxidant like DMSA or DMPS, one of these things, and that kicks it all out. But even that doesn't work well. That doesn't work well because 95% of all of your toxics, whatever lead or cadmium is leaving your body, 95% of it leaves in the stool, and only 4 or 5% in the urine. So what they're doing with these challenge tests, and these are very popular, a lot of people doing this, they're actually forcing all those toxics out a different door. So any human being, we all have some lead, mercury, and stuff in our bodies. Anybody who does the challenge test, you'll probably see 10 or 20 times more in the urine, and the doctor might say, "Oh, look at all the mercury we took out," and then they'll do that challenge test again in 3 months, and hey, we got some more we got to get out. I've seen people do that for 3 years. The half-life of mercury in the body is 42 days. It's all gone. But they're getting improvement because these are great antioxidants, and they're getting symptom improvement because of their great antioxidants. Unfortunately, they only last about 17 days, so the improvements fade away.
Interesting. So it's probably that these are great antioxidants, and it is detoxing mercury. If you happen to be sensitive to mercury, or if you're just lowering the oxidative stress in your body by detoxing heavy metals, it's probably like a chronic treatment that some people may want to do, but it's not actually measuring how high your burden is for heavy metals.
Not at all. And another thing is it can cause problems. For example, cadmium. If a person, some people have quite a bit of cadmium in their body, surprisingly how many. If you quickly push a lot of cadmium out through your kidneys, you will get kidney damage. That's been known for 50 years. And the way it's supposed, the cadmium is supposed to leave is through the stool. So you actually there's some risk involved with doing it with the DMSA and DMPS, EDTA. They're great if you've got we've had patients who were actually poisoned with mercury or lead, and we just sent them to the hospital and did these chelation tests, and it only takes like 4 or 5 days, and then the patients are fine. It only takes 4 or 5 days.
Interesting. So, how are you testing neurotransmitters in the body? Do you do a urine test for neurotransmitters, or are you measuring them indirectly by symptoms or cryptopyroles or serum zinc, or more like plasma zinc? Sorry.
It's a sad story. It's a sad story. I was all excited about this, and I had researchers that were studying serotonin kinetics in plasma and in membranes, and I did this for years, and I've given up because I'm convinced it's quite meaningless. It might have a lot to do with your GI tract and your liver, but not your brain. So for mental patients, it is quite worthless. For example, serotonin. Most of our serotonin is created in our GI tract. Okay, that's where most of it's synthesized. None of it gets to the brain. Absolutely none. There's a lot of people who are analyzing serotonin in the body and trying to make, therapy decisions. No, the serotonin that's synthesized in the brain is synthesized in a different way. And so the problem is
that's going to disappoint a lot of people. I mean, like, there's so many people who get these urine tests and this test, and I hear my neurotransmitters, and often I'm just like, I don't know what to make of these tests, really, because, unless they do surgery in your brain, I don't.
Yeah. You're absolutely right.
What do you do? You look at it by the symptoms?
You might get some validity from GI tract studies, because if your serotonin is screwed up in your GI tract, that you might be able to help the GI tract, but not the brain. And we have to do this. Fortunately, the symptoms of high dopamine or low dopamine, et cetera, they are quite well known. Psychiatrists know this, and when they treat with these medications, they're looking strictly at the neurotransmitters, and most psychiatrists are quite good. I mean, if you've got a person who has panic disorder, or if they have epilepsy, that has to do with hyperactive neurons. So they're going to want to quiet down the neurotransmission at places like norepinephrine, and that's what their drugs do.
So you're basically looking at it based on the symptoms. You're not looking at it based on any objective test. It's just like, okay, if you have these symptoms, if you have panic disorder, you probably have overactive glutamate, right? Or maybe dopamine as well.
Well, yeah, you're partly right. The reality is that there are chemicals, there are lab tests we do that are associated with these very neurotransmitter abnormalities. So we do both. We look at the symptoms. I think 50%. It's really a 50-50.
So which lab test would correlate with a neurotransmitter, for example?
okay? If you're undermethylated, you probably have elevated norepinephrine activity. Same is true if you have high free radical copper. That is, if you've got a lot more serum copper than you do ceruloplasmin. We know that if you have zinc deficiency, you're likely to have a pyrole disorder, and that's a recipe for low serotonin activity. It's also a recipe for low levels across the board of some of the neurotransmitters. So it's I think 50% of our diagnosis is based on lab work, 50% on the medical history and the symptoms and traits. And thank God we have the really classic sharp differences in human beings that people are born with. It has to do with the methylation that occurs in the 9 months of gestation. When we are born, our DNA has these methyl and acetyl bookmarks that are in place, and they're usually there the rest of your life. And that has a lot to do whether a person is competitive, whether they have allergies, etc. It has to do with genetics, and also has to do with the environment in the womb while that child is being created.
Interesting. One concern that I have is that when you deal with something like dopamine, right? Yes. It depends where the dopamine is. For example, if you have high dopamine in your striatum, then that's like, first of all, that can lead to schizophrenia, but in general, it could also lead to higher levels of motivation, right?
It's really
it could lead
higher dopamine neurotransmission. It's not so much the amount of dopamine, it's the neurotransmission.
Sure. So, okay, that's a good point. So if you have higher dopamine neurotransmission, because I am aware you have the DRD 1 to 5 receptors, you have the dopamine transporters, you have a whole bunch of dopamine related neurotransmission proteins.
Yeah, 12 the serotonin receptors, right?
Exactly. Yeah. No, I'm aware of that, and so. Okay, so but I was kind of trying to simplify it for the people like
sure.
Dopamine, but you're right. The whole neurotransmission of it, you could have high neurotransmission in your striatum, and that will cause higher motivation, higher pleasure, let's say, rather than if you have higher transmission in your prefrontal cortex, it might contribute to anxiety, let's say.
Yep.
Right. So it also depends on where it is, the neurotransmission of dopamine or serotonin. In one place in the brain, it could be good. In one place of the brain, it could be bad. So how do you account for those variables?
Well, first of all, you're pointing out one of the most remarkable aspects of human existence, and that is that during the 9 months of gestation, your body is tailoring the expression of these things, including the dopamine capability. In every cell line and every tissue is different. It's beautifully programmed. But like you're pointing out, for some people, the striatum of the brain might have too much or too little dopamine. And it'd be nice to find that out. And symptoms really help with that. You just pointed out the symptoms, some of the major symptoms you get if it happens to be in your prefrontal cortex or in the striatum. So yeah, we are able to use the symptoms together, hopefully with methylation and epigenetics, and get a pretty good idea. It's not perfect, but it's usually we can confidently treat people. And
higher dopamine transmission in the striatum also causes anxiety?
Not as much of the prefrontal cortex.
Oh, I see. Okay.
Not as much.
I thought it was I thought it was mainly in the prefrontal cortex.
I would like to say one thing that people that would like to dive into this. I did write a book on the subject. I wrote it 7 years ago, but it's still really applicable and can really, if people want to dive into this whole area, it's called Nutrient Power. It's available on our website. That's something somebody can do if they're interested in learning more about this. Also, our website itself has a lot of information. It's walshinstitute.org. Thank you for showing the book.
So here's the book, Nutrient Power. I did read some of it. I didn't get a chance to read all of it, but it looks the part that I did read was very interesting.
And if somebody wants to really dive in, they can get the book from our website. You can also get it on Amazon. It's been an Amazon bestseller. But if you buy it from our website, it's about the same cost, and 100% goes to our charity for research, et cetera. Anyway, I think that a lot of these concepts are somewhat new, but we are now training doctors. I've got a team that travels around the world, and it's just growing and growing. in April, we'll be training nearly 200 doctors, many of them psychiatrists. And what they tell me, after we've completed this training program, it's an intensive training program, and show them how to do exactly this, most of them tell me that it's changed their lives and the way they treat patients, and that now they can help patients they couldn't help before. They say may still use medications, but now they've got another weapon in their arsenal. So that's growing and growing. I think this is what people are going to do in 20 years. They're going to do what we do. And
that's super interesting. I mean, I definitely want to do more reading about the NMDA receptors and various conditions. Okay, thank you so much, Dr. Walsh, and I appreciate you spending the time and being enlightening for a bunch of different topics that I had some preconceived ideas about.
Well, you've asked really good questions, and it's been a pleasure talking to you.
Okay, thank you so much.
Great, you're welcome.
All right. Okay, bye-bye.